So, welcome back. We are in Bio 212, Anatomy and Physiology 2, and we're working on Chapter 28, Part B here. And we're moving on to the hormonal control of the testes. How do you regulate spermatogenesis of the spermatocytes from spermatogonia as a result of changing concentrations of hormones, right? So, way back when, inside Chapter 18, you learned that. Let's take a pen here. You have a central nervous system. Here's your brain, right? Here is your pons, your medulla, spinal cord. Hopefully yours doesn't look like that. And your cerebellum is back here. And here's your infundibulum. And you have an anterior pituitary gland and a posterior pituitary gland, right? And at puberty, what's going to happen, of course, is gonadotropic releasing hormone is going to be released from here and it's going to target the gonadotrophic cells on the anterior pituitary gland. And that's going to cause the release of follicle-stimulating hormone and luteinizing hormone right here. What happens, of course, is they use that hypophyseal portal system that you've long since forgotten about, I hope not, and they're going to target the actual population of cells inside the testes that are called Leydig cells. And those Leydig cells will now take cholesterol, okay, and convert it into testosterone. Okay, and they're going to what ends up happening is the production of testosterone itself ends up decreasing the secretion of luteinizing hormone and gonadotrophic releasing hormone. So its own product, and the way your textbook does this is it makes it a series of dotted lines. You should pay attention to when you're looking at figures, what the solid and the dotted lines mean. What's going to happen, of course, is that I should say, in this case, in puberty, right? There's an enzyme, 5-alpha reductase, which is going to convert testosterone into dihydrotestosterone, which is going to now allow for the development of external genitals as well as the prostate. Okay? So this is an introduction. It's pretty straightforward, and as long as you remember the material from Chapter 18, you're going to be fine. There'll be some more details as we go through this, but this is what the luteinizing hormone is doing here. Luteinizing hormone, okay? Now from that trophic cell targeted to Leydig cells. What's follicle-stimulating hormone doing? Well, it's indirectly regulating spermatogenesis because what's going to happen is follicle-stimulating hormone is going to act on those Sertoli cells. Remember those sustenticular cells or nurse cells? And they're going to make this thing that's called androgen-binding protein. And what's significant here, of course, is that I get to stop for a moment and say, you remember that cholesterol is the building block for testosterone, so therefore it's not water soluble. And that means the androgen binding protein is responsible for increasing the solubility of testosterone. Okay? It's the testosterone that ultimately stimulates spermatogenesis inside the testes. Okay? So without androgen binding protein, small levels of spermatogenesis here. Okay? Sertoli cells also release another hormone, okay, and that's called inhibin. You're going to see both males and females make inhibin. They have slightly different responsibilities, but generally speaking, inside the male, you're going to have inhibin is going to release, or I should say, inhibit follicle-stimulating hormone. And if you just work through the logic quickly, well, follicle-stimulating is responsible for the production of androgen binding protein here. So if you lose inhibin, therefore you lose this. Therefore, it doesn't matter how much testosterone you make. It won't get to its target. Okay? So that's a good place to pick up when we're thinking about the hormonal regulation inside the body, right? So we'll go through a cartoon of this. Unfortunately, I have to get rid of all of this to change the slide. We go like this. And inside your textbook, you should see a cartoon that looks something like this. And again, here is your hypothalamus. I could draw that cartoon again for you over here, but you can see it all right here. Here's the hypothalamus. Here's your anterior pituitary gland. Here's your posterior pituitary gland. This is a good opportunity to make sure you pay attention to some really obvious oversights by the artists who put this textbook together. There's no line that's actually going through the posterior pituitary gland. I think you get that, but I just want to make sure. Gonadotrophic releasing hormone targets the gonadotrophs. They release follicle-stimulating hormone and luteinizing hormone. That follicle-stimulating hormone targets the cells over here. And those are, of course, the spermatogenic cells. Okay? What's going to happen is the Leydig cells are making testosterone, so the testosterone targets to create spermatogenesis over here, but because the follicle-stimulating hormone is now allowing androgen-binding protein, what's going to happen is it can travel throughout the rest of the body. Okay? Something that I didn't mention before, but I want to make sure you pay attention to. Both testosterone and DHT, okay, increase anabolism. Okay, and inside your little purple part of this cartoon, it says protein synthesis. Well, protein synthesis means many different things. So let's switch over to this. Let's go over here. Let's make sure you get that. Protein synthesis requires amino acids, right? A. Doink, that's an A and an A. For those of you who are whining about my handwriting, I apologize ahead of time, but that's it. Okay? The amino acids have to be brought inside a cell. To put amino acids together to make a protein, we need to drive transcription and translation, TX, TL, right? And this is what I'm thinking when I look at anabolism. How much energy is required to do all of this? Not just protein synthesis. You need to break down more glucose for that to happen. So every time you're thinking about what hormones are actually doing, you should be thinking, are they increasing anabolism or are they increasing catabolism? Okay. So let's get rid of that. Make sure you now that we've actually made our products, notice that there's a series of red lines. The testosterone targets both the anterior pituitary gland as well as the hypothalamus, where follicle-stimulating hormone only targets the anterior pituitary gland. And that may not seem like a big deal to you, but, okay, this is the sort of stuff where Mother Nature has obviously engineered a fair amount of regulatory control here. So you would say that the hormone needs to be more tightly regulated than the binding protein or the inhibin that's made as a result of this. Okay? Does that make sense? Cool. So let's get out of here. Let's go down here. And this is essentially a retelling of that cartoon. Okay? Where you can actually see some stimulus is going to disrupt what's going on, and the hypothalamus is going to increase that in, say, the youth, male or female. Some visual hormonal stimulus is going to change this. And you can see if you actually have the negative feedback system here working properly, you no longer need a high level of, in this case, testosterone, right? And what's going to happen is the amount of gonadotrophic releasing hormone is decreased, and therefore the anterior pituitary gonadotrophs are decreasing the amount of luteinizing hormone. And because there's less luteinizing hormone, there's going to be less testosterone. And because there's less testosterone, there's not as much need for the androgen binding protein, or for that matter, inhibin. So again, I'm just reinforcing the fact that Mother Nature wants to make sure that you understand that we need to regulate the production of the hormone because it's going to take the most energy. Okay, so now we actually have to get these materials out of the actual male body itself, and that means we actually have to look at a system of ducts. And I cannot stress this enough. You're going to take this for granted. You think it's going to be pretty straightforward, but if you put these out of order, Mother Nature won't let you let material out of the body. Okay, there is a process by which we're going to have to increase the amount of pressure inside of these ductal systems, right? It's a pressure generated by the fluids that are produced by the Sertoli cells as well as the other glandular structures. Seminiferous tubules are going to travel into the rete testis, which is now going to push along. Once inside the epididymis, it's a series of tightly coiled cells, but what's going to happen here is we now have a modification. That modification is a stereocilia. You should be able to see this inside lab when you're actually looking at the slides. That's how you differentiate between being inside the actual seminiferous tubules versus the epididymis. This is where maturation will actually occur because now the tails acquire their ability to become motile. They literally start flapping their tails, okay? And they can stay there for months. In fact, if they never leave the epididymis, they're atreuged. They're broken down and they're turned back into all their basic building parts, Okay. However, if they do move, they travel through the vas deferens, which is now going to allow material to pass as a result of sexual arousal out of the body. They can also stay here for several months as well. Again, the same mechanism in place can break all of this down. It's called atresia, where literally the entire structure is disintegrated and everything from the single-strand haploid DNA to all the glycoproteins to the mitochondria, it's all just broken down and turned into building blocks. Okay, so if it's going to get out, you're going to see it's going to travel through a spermatic cord. You're going to have ejaculatory ducts, and then finally it exits through the urethra. So let's just show that to you here. So this is the posterior aspect you're actually looking at here, where it's traveled through the vas deferens. You can now see it's passing through this here's a seminal vesicle here. What's going to happen is materials made here will travel, be collected. This entire structure is going to mix together. Materials made inside the prostate here, okay, are going to be mixed as we actually now travel into the urethra. And you're going to see there's different names for the intermediate urethra and all of that. And I suppose that's important as long as you understand that it's a tube, and the tube has a lumen, and the lumen has now a volume of fluid inside of it, and all of those fluids have to mix. This is the base of the penis here, where you actually have these things that are called the crew, and this is the bulb. And what's going to happen is, upon stimulation, and we'll learn how stimulation occurs in a few minutes, where what's going to happen is you actually push the back of the penis into the pubic bones of the male, and that's what's going to allow for the change in the shape of the penis itself. And in doing so, it allows the urethra to be opened. Okay? So what's actually going on here? What are we mixing in? Okay? So you can see here that we have these accessory glands. Okay? And you have different parts. The seminal vesicles, okay, make up about 60% of the volume, okay? And they're secreting an alkaline viscous substance that it contains fructose. Notice this is not glucose. This is the only, how do I say this? The only part in the body that doesn't primarily use glucose as its carbon source. You have prostaglandins, which are going to create temporary inflammatory responses as well as smooth muscle contraction, and you have clotting proteins. So they're going to mix throughout all of the spermatocytes, so at ejaculation, depending on where it happens, you're going to now allow for all of those to clot together. These are going to be different from clotting proteins we're going to talk about inside chapter 19. Prostate makes 25% of the semen volume, okay? And you're going to see here that it's milky, slightly acidic, containing citric acid, fructose. There's a different energy source. Citric acid goes right into the Krebs cycle, which allows us then to begin utilizing energy. Many proteolytic enzymes here, which means we're going to have materials that can allow not only for the breakdown of the clotting here, but also materials that can be encountered inside the female reproductive tract. It also comes with its own antibiotic. Okay, so the prostate makes seminoplasmin, which is now going to help the semen survive the travel, number one, but also two, the location where fertilization can occur. Finally, we have the bulbourethral glands. And these are the hardest things to find inside lab, okay, but they're generally speaking there where the bulb of the penis is. You might remember where I showed that to you a moment ago. Okay, it makes an alkaline fluid, and what it does is it neutralizes the acids alongside the urethra, and your textbook says it lubricates the end of the penis and the lining of the urethra. What it's actually doing, I wouldn't say lubricating is the word, right? It's actually reducing resistance, meaning a fluid now occupies all parts of that urethral lining, so that when materials are forced out rapidly, there's no resistance and there's no clotting that would actually happen inside the male reproductive tract. So how does it work? Well, you're going to mix all this material together, right? And you can have anywhere between 2 and 5 mils of semen, which is the ejaculate. And your textbook gives you this value here. Everything I've ever read outside of this has been anywhere between 200 and say 350 million. Okay? But that doesn't matter. It just takes one. But it's a numbers game. Okay? So I said it 2 buts there. It's a numbers game to see which is the one that's actually going to make the trip properly. It's slightly alkaline, okay? And what's going to happen is the materials that were made from those accessory glands are going to allow for the actual protection and traveling of the semen because it's going to coagulate after ejaculation. So how does it actually happen? Well, you should probably rearrange this a little bit. For erection to happen, right? How do you actually get the male reproductive structure to become changed in structure, right? Parasympathetic fibers cause the local release of nitric oxide. Again, chapter 18, but also chapter 15 inside of your textbook, causing smooth muscles inside the arterioles to relax. Okay? So, parasympathetic nervous system has to become activated first for blood to move into the actual spongiosum and cavernosum. We'll come to that in a second. Now what's going to happen is the materials after erection has happened, you now changed the pressure on every other part of the penis. And that's what's actually going to happen. That gland's penis is actually going to become enlarged as well. And the Pacinian corpuscles inside of the gland's penis are what actually now allow for the activation of the sympathetic nervous system. Okay? So you got to read through that closely inside your textbook to figure out that parasympathetic actually has to happen first. Okay? So let's take a look at what's actually going on here. Here's that cartoon where I showed you the bulb with the penis. That's where the bulbourethral gland is going to be. Materials are moved here. If this is an unerect penis here, at the activation of the parasympathetic nervous system, nitric oxide is now going to move to the vasculature here. The smooth muscles will relax inside the vasculature, and now all of this becomes engorged with blood. Notice here is that spongy urethra, okay? And here are the corpora cavernosa. Mother Nature ensures that, remember, the bulbourethral gland has made that now material to coat the lumen here. And what's going to happen is, as this gets filled with blood, the corpora spongiosum gets filled with blood as well. And this now will allow for a path, a conduit path, to get now the spermatocytes out of the body. Okay, so in this second part, what we did is we spoke about what are the hormonal requirements for the male reproductive system to function properly. And then we turn to, well, if you make spermatocytes and it's regulated hormonally, then where do the spermatocytes travel to? Meaning you have to engineer a system that will allow the spermatocytes to meet a secondary oocyte. So your chapter inside your textbook finishes off with this description of what is required at the endocrine level, meaning you have to activate the parasympathetic nervous system to allow blood to flow into the penis, to now allow for stimulation by the sympathetic nervous system, which will then cause rhythmic contractions of muscular structures to now push, because remember, it's a fluid pressure system here, which will now push the fluid out of the body itself. Okay, so that's a pretty good place to stop. And what we'll do is we'll now move into the female reproductive system. Okay, so in the next set of discussions here, or introductions, okay, what we're going to do is we'll talk about how does the female reproductive system differ. Okay, speak with you in a few minutes. Bye.